In vivo gene therapy involves DNA modification in cells within a patient's body. One such therapy is personalized for the baby KJ Muldoon, who suffers from the genetic disease carbamoyl phosphate synthetase 1 deficiency. CPS1 deficiency causes the accumulation of toxic levels of ammonia in the blood, as CPS1 expressed in the liver transforms ammonia into urea. The less toxic urea is excreted in the urine. The base editing therapy for baby KJ Muldoon uses an mRNA encoding an adenine base editor and a guide RNA targeting the Q335X mutation, which is a G2A stop variant. The mRNA for the editor and guide RNA are carried by lipid nanoparticles and administered intravenously. LPNs can be designed to target receptors on liver cells, ensuring liver cells and not other cells specifically take them up. In the liver cell, the adenine base editor expressed is directed by the guide RNA to the location on the DNA sequence where the precise base substitution is required and reverts the mutant allele to a wild-type allele. By May 2025, 3 treatment doses were delivered, and baby Muldoon showed signs of clinical benefit. Then there is ex-vivo gene therapy, which involves removing cells from the patient, modifying, and then returning them to the patient. An example is Casgevy, which treats sickle cell disease and beta-thalassemia. Both diseases stem from gene mutations that code for the beta-globin subunits of the hemoglobin of red blood cells that carry oxygen so vital to our survival. In this therapy, the patient is treated with the chemicals to stimulate the bone marrow to release stem cells into the bloodstream, and these peripheral blood stem cells are collected. Electroporation will infuse the Cas9 and guide RNA into these stem cells. Cas9, directed by guide RNA to the BCL11A gene, causes double-strand breaks, triggering the cell's DNA repair mechanisms, which generally generate mutations, knocking out the BCL11A gene. Once the patient has undergone conditioning therapy to clear out existing bone marrow and blood stem cells, making space for the modified cells, the gene-edited cells will be infused into the patient intravenously. The stem cells travel through the bloodstream and settle in the bone marrow. Since the BCL11A gene function is disrupted, the body produces fetal hemoglobin with gamma globin subunits instead of adult hemoglobin with the variant beta globin subunits, thus alleviating disease symptoms. Yeah!